And now, Amy, we are very excited about your talk on your mouse models.
Yes, perfect. Let me see. This is the one I need. So again, thank you all for being here today. My name is Amy van Hattem. I am a medical doctor and a second-year PhD student in the lab of UBE3A. And I work in the Erasmus Medical Center. Today we've already heard two of my colleagues talk about the clinical work that we do there. But really one of the core strengths of the Encore Expertise Center is also the collaboration with fundamental research. And in our lab, we work on several neurodevelopmental disorders, but we have a very specific interest in Angelman syndrome and Dup15q syndrome. Let's see. Yeah, so there are basically two very, I think, simple but very powerful insights about Dup15q. The first one is that, let me see if this works. It matters who you get it from. So we get one chromosome from our father, one from our mother, and we know in case of Dup15q syndrome, the duplication needs to be on the maternal chromosome, meaning that also patients with the paternal duplication really present in a different way, often more mild. I think a very clear example is the fact that some mothers carry the same duplication, but then on their paternal chromosome. But once they give it to their child, who then receives it on their maternal chromosome, they show the disability. The second one is that gene dosage matter. So we know that as the duplication size increases, but more often that you have more copies of a certain gene, this also correlates with the disease severity. So yeah, how did we get there? But mostly, how does that help us find or think of new research questions? So a Dup15q, and I know this gets a bit more into the fundamental side, is a copy number variation. And these are structural rearrangements in our chromosome or in a genome that can lead to either extra or missing copies of DNA. And it happens at regions that have a lot of repeats in their structure. And during a process called non-allelic homologous recombination, and please all forget that word, that will exchange parts of the DNA, which then could potentially lead to a deletion or a duplication of part of the DNA. This is not, in fact, always bad. It's very important for us, also in evolution, but some of them are pathogenic and they can be associated with disease, such as is the case with Dup15q syndrome. Now, I would like to zoom in a bit. So we are talking about chromosome 15, and here you see an image of the region that we're talking about in mature neurons, so in mature brain cells. And we know this region is subject to genomic imprinting. And imprinting means that some of these genes are only expressed on, for instance, the paternal allele. So here, those are indicated in blue. They're only expressed on the maternal allele, so that's the one in orange, or they're expressed on both alleles, here in green, and we call those non-imprinted. And then it matters where this copy number variation takes place. So if there's a deletion on the paternal chromosome, this gives Prader-Willi syndrome. If there is a deletion, well, there are different mechanisms, but in general, if there's a deletion on the maternal chromosome, there's Angelman syndrome, which is a term you've already heard a couple of times, I think, today. But then if there's this duplication, we are talking about Dup15q syndrome. So it happens who you get it from. The second lesson was that the gene copy number matters. So we know that interstitial patients have an additional copy of the genes in this region, and then the IDICs have even more. And this again correlates back to the symptom severity. And ultimately the question is what genes really drive this syndrome. And our initial hypothesis was that UBE3A would be the main driver, which sounded very logical to us because it's the only gene that is maternally expressed. So that's the orange gene you see over here. But then if we really look at the evidence, there's only a single family that is described with a microduplication of UBE3A. And this family showed a milder phenotype. But then also there are a few patients that have a UBE3A gain of function mutation, so also a more active protein. But then again, it's a very heterogeneous group. So then we tried to answer this question in the lab. So we created a mouse model that had overexpression of UBE3A, only of UBE3A. And we did see that there's an increase in protein levels in these mice. But then when we tested behavior, we did not find very significant differences So from this study, we concluded that only UBE3A overexpression was not enough to cause a Dup15q phenotype. So then the question really became what other genes could potentially play a role. And here I think you can already see I highlighted some of the genes in the region that have already been associated with neurodevelopmental disorders. So you have the GABA receptor subunits, they're involved in early onset epilepsy, but also some of the others are associated with intellectual disability or autism spectrum disorder. So what genes really do play a role? We are taking several approaches for this. So one is from more of a clinical perspective. So we're also, and I think this kind of correlates with the story that Dr. Nevado recorded in his presentation. We're really looking for more of the atypical duplications, so the ones that are actually smaller than the ones we would usually see. And we are trying to find the duplications that are enough to basically cause a Dup15q phenotype and enough to, have the kids diagnosed and need proper care. And we're really trying to find like the smallest region that needs to be duplicated to then discover the genes that are really involved. So if you have any patients like this, we would love to collaborate in this. I now know who to reach out to as well. But the other part is really more mechanistically. So we want to study the role of UBE3A and the non-imprinted genes, so the green genes. And basically we're trying to dial the system and upregulate UBE3A or these other genes and then see what happens. And the model that we decided to use for this is a mouse model. And the reason for this is that ultimately we want to find a model that really represents part of a population. So we want to find a model that represents Dup15q. And that allows us to then start to think about what goes wrong mechanistically, but then also use different tools to find new therapies and bring that back into the clinic. And there are several important requirements for a model system, and specifically for mice. And I would like to highlight the top two of those. So first, we want to have construct validity, and that's something that means basically the model should have the same genotype, so the same genetic alteration as the patients. Well, the nice thing about mice is that they actually have a quite similar brain to humans, so both on an anatomical level, but also cellular and on a molecular level. And this really allows us to study brain development, also at very young ages. Also how these neurons, so the brain cells, connect and how they communicate with each other. And actually, if you look over here, and I think you can appreciate that this region of mouse chromosome 7 is very similar to the picture I showed to you earlier, where all the non-imprinted green genes are clustered together. So really, this really helped us in making a new mouse model, where we created a interstitial duplication using a CRISPR-Cas9 system, where we have an overexpression of all of the green genes, so all of the non-imprinted genes, without a functional UBE3A copy. We validated the system, so we looked at mRNA levels and protein levels to show that indeed we have this upregulation. But then really I want to focus on the next thing. So what we do is we cross mice with only the overexpression of these non-imprinted green genes, here also in green. And we cross them with a mouse that has only the overexpression of UBE3A, which is the mouse model I talked about previously. But that really gets us to 4 groups of mice. So wild-type mice, WT, is a mouse with the normal functional amount of copies. You have an overexpression mouse in orange with only overexpression of UBE3A. You have a mouse in green with only the overexpression of these non-imprinted genes. And then you have a mouse model with both UBE3A and these non-imprinted genes that are overexpressed. And this last mouse really correlates well with the amount of copies that we see in patients that I indicated in purple. So the next thing we really want to look for is phase validity. And phase validity means that these mice should show the same phenotype or behaviors as we see in the patients. So actually we start in the patients. So we start to see what symptoms do they have. So do they have intellectual disabilities, motor problems, or sleep problems? And we link that to behaviors that we can test in mice. And mice are actually have quite a rich repertoire. So there's a lot of behavioral tests that you can do. Some of these behaviors translate really well to human behavior, but also specific brain regions involved. Other behaviors are more species specific, so mice build nests, we don't do that. But they do tell something about UBE3A levels or treatment effect. So for this mouse model, we also did a couple of behaviors, and I'll highlight a few of them. So the first one is a rotarot. Basically, in this test, the mice learn to walk on a wheel that keeps on turning and goes faster and faster. And you measure the time until they fall off. And you do this over multiple days. And as you can see in the graph, we did see a difference on day 1. But then as they start to learn this task, they all do it in a similar fashion. And on the last day, we did not find any differences between the 4 groups. We also did a float or swim test, which is a behavior that doesn't translate well to human behavior. But we know it's very sensitive to UBE3A, also based on work we did with other mouse models. And we did indeed see a very clear effect of overexpression of UBE3A, but there was no extra effect if these non-imprinted genes are also duplicated. And then finally, because we know that autism spectrum disorder is quite prevalent, we did a social behavior task, which is called the 3-chamber. And we did find some interesting differences in the time they spent in certain chambers, but then really looking at time interacting with each other, we did not find a difference between the genotypes, but it's something we're still looking into and seeing if we can do additional tests to really get this clear. We are not finished yet, so we're still doing quite a lot of tests, either for more of a learning paradigms, but also epilepsy, which is something we're very much interested in. We're also going to do EEG on these mice and look at sleep, so their sleep rhythm, to really figure out if this model is suitable for further testing with, for instance, treatments. But then also we still have a lot of ideas, so we can still change the model, have even higher copy numbers. But then also depending on the information we get from the clinic, we can start to specify the region that needs to be duplicated. And with that, I would like to, I mean, these are very general take-home messages, but I think this really shows the strength of also trying to figure out mechanistically what goes wrong in a syndrome or in a disease, because it really allows for, the increase of information really allows for more understanding, and then also finding the therapies, gene therapies, and explaining what we see in the patients. Again, contact us, and this is the lab that I would really like to thank for all of their help.
Thank you, Amy, to bringing us this very complicated topic so clearly and explain to us. Are there room for 1 to 2 questions?
You were saying you wanted smaller duplications, but you don't want micro duplications.
Well, our initial hypothesis was, of course, only UBE3A would be necessary, which is something I think at this time we no longer believe. In the patient data set that we have right now, we're actually surprised to find some patients, for instance, that have like HER2 or some of the GABA receptors possibly not included within the region, but there's still like enough, they still show enough phenotype to require genetic testing, and then also they have phenotypes enough to that require medical support. So we're really looking for patients that have, most likely anywhere within the region of BP2 to BP3, duplications that are just smaller. Yeah. And I think Sebastian also might have some very nice findings in that one.
Yeah, I have another question. Maybe to look at the different level of phenotypes, so not the behavior, but more molecular phenotypes. Have you ever thought about doing some, mRNA sequencing or proteomics to directly see if really the copy number changes have results in gene expression and also maybe tissue-specific gene expression?
Yeah, so for this mouse model, we did RNA sequencing, although on a bit of a smaller number of samples. So we might want to do another analysis, and then we're also doing proteomics We happen to have done it, but then something technical went wrong. So we're adding the proteomics as well. We do have all this data from the overexpression mouse model already, but it would be really interesting to see if the double mutants, so the one with both UBE3A and these other genes overexpressed, if that changes the transcriptomics. Yeah.
We have collected for patient organizations. We know we have like 10 that have only 1.2 and only 3.2. So the edges, we actually have them also in the patient organization, because I think that would be something. And some of them are in the registry, not all of them.
Yeah, it would be quite interesting if there, because I think I also saw on your data set patients within like only BP1. Oh, you don't have BP1, I think, but because all of our patients have at least UBE3A included in their duplication, which we think is at least the minimal gene necessary, since it is a maternal duplication syndrome. But it would be really nice to put everything together, and that's really what we're hoping for in this study.
So we have some interstitial breakpoint 2, breakpoint 3 patients, smaller, so below breakpoint 3, more proximal of breakpoint 2. I think there we don't have any patients.
Yeah, we have quite a few patients with breakpoint 1 also included.
Yeah, but also the breakpoint.
Yeah,
Yes.
yeah.
So you really have the breakpoint 2, 3 as kind of minimal region.
Yeah, exactly.
Probably the other ones don't have a phenotype.
Yeah.
Don't get tested.
I was wondering whether do you think that you'll be able to develop a sort of epilepsy model of both a UBE3A only duplication and the full blown duplication to see possible differences between these two situations?
Yeah, so for the mouse model with only UBE3A overexpression, we know they don't have any spontaneous seizures, which is something you see more often in mouse models. Depends on the background as well. We did a inducible seizure test with a loud sound, which we know works very well for Angelman syndrome mice, but we found no differences in the UBE3 overexpression mouse. We're now doing, and that's what I'm going to do this summer. It's called a fluorotholsin, a different inducible seizure test, and we expect to find differences there because we have done it in the overexpression mouse, and actually we see a lot of CREP in those mice. So it would be really interesting to see how that works in this mouse model. Yeah. So I think we'll get some nice findings in it. Yeah.
Okay, I'm very happy about this discussion now, but we have to move forward. Let's talk about this, discuss it later. Okay, thank you, Amy.